I'd like to introduce myself at first. This is Roxana Parveen, Senior Lecturer, Department of Biology, Maristone College. I'd like to tell you something about today's class. Today we are going to discuss about immunology. Yes, the very important topic nowadays, because we need to at first look after our health and then we can think of anything else, right? So let's go forward. Well, today's topics are roles of vaccines in immune defense, concepts of vaccination, and roles of memory cells in immunity. This is the diagram of a vaccine which is injected to get rid of diseases in advance. So let's know what is vaccine. A vaccine is a substance that is introduced into the body to stimulate the body's immune response. It is given to prevent an infectious disease from developing and the person becoming ill. How vaccines are made of? Vaccines are made from microbes that are dead or inactive so that they are unable to cause diseases. I would like to add one more point, that when we insert the microbes in different forms or inactivated forms, our body can create the immune defense in advance by recognizing the antigens. However, a vaccine may contain live attenuated or killed microorganisms or parts or products from them capable of stimulating a specific immune response, which are comprised of protective antibodies and T-cell immunity. A vaccine should stimulate a sufficient number of memory cells and B lymphocytes to yield effector T cells and antibody-producing B cells from memory cells. There are different sorts of vaccines though. The viral vaccines should also be able to stimulate high titers of neutralizing antibodies. Injections of a vaccine into a non-immune subject induces active immunity against the modified pathogens. Let's discuss about different types of vaccines. There are many types of vaccines. Among them, at your syllabus, you may have to describe 5 types of vaccines. One is live attenuated vaccine. Some vaccines contain live attenuated microorganisms. Many of these are active viruses which contain virulent properties to produce a broad immune response. Some are bacterial in nature, such as viral vaccines are for yellow fever, measles, mumps, and other bacterial vaccines for the diseases typhoid. Mycobacterium tuberculosis vaccine contains a virtually modified strain called BCG used to elicit an immune response to vaccine. What is inactivated or killed vaccine? Some vaccine contains killed but previously virulent microorganisms that have been destroyed with heat, radioactivity, chemicals, or antibodies or antibiotics. These including influenza vaccine, cholera vaccine, plague vaccine, polio vaccine, hepatitis A vaccine, and rabies vaccine. Subunit or purified vaccines. Certain subunit or fragmented protein can create an immune system in many cases or in many causes, like subunit vaccines against hepatitis B viruses are composed of only the surface proteins of the virus. The virus, like particle vaccines against human papillomavirus, is composed of the viral major capsid protein and the hematoagglutinin subunit of influenza virus. You can get these ideas from your textbooks too. What are the toxoid or inactivated viruses? Toxoid vaccines are made from inactivated toxic substances which cause illness rather than the microorganisms. The toxoid vaccines includes the tetanus, diphtheria, and so on. All these diseases, are actually not compatible to this vaccination process. For though we can put an example like Crolitus atrox toxic is used to vaccinate dogs against the rattlesnake bites. Though these are not so much important to know, you have to describe different examples of the toxoid like tetanus toxoid or TT and diphtheria toxoid. Okay, and last not the least is conjugate vaccine. Certain bacteria have polysaccharide outer coats that are poorly immunogenic. By linking these outer coats to proteins like toxins, the immune system can be developed. Let me clear about this concept. What is poorly immunogenic? The outer coating of this sort of polysaccharide bacteria cannot trigger the presence of any antigens or any protein by the complement protein. You know what is the set of complement proteins? They can trigger the overall chemotaxis process. Okay, by the linking of these outer coats, of course, is very important to have opsonization process. As in case of this vaccine used to Haemophilus influenzae type B vaccine. Okay, so these are the types of vaccines. There are other less important vaccines. We can mention that dendritic cell vaccines, DNA vaccines, and so on. In this part of the lecture, we are going to discuss about the memory cells. Do you know what is memory cells? Do you have any experience of having pox or sort of mumps, rubella sort of diseases second time in your life? Probably no. In maximum cases, they don't repeat because our body recognizes these sort of viruses as their antigens, and they create memory cells inside the body. So what is memory cell? This is not the hard drives like the computer. This is not the pen drive to store the data. These are more amazing than that. Okay, so what is memory cell? The immunological memory is a unique property of the immune system as it can store the information about a stimulus and can mount the effective response when the stimulus is encountered again. That means, to be very simple, immunological memory cells are the very unique property which can recognize, which can set up the memory about the killing of the germs, the mechanism of the phagocytosis process and others when they experience the exposure of the same germ again in the body without letting the person or the individual know that they are being exposed, right? So this is amazing. Yes, let's see the diagram. Okay, you see in this diagram, this is the B cell, right? The very important part of the immune response. These B cells are creating some hook-like structures over there, you see, and the green colored substances are showing the bacteria. When they get attached with the bacteria, this one is the first, okay? It's just screaming like that. And secondly, the B cell clones. How? At first, this B cell attracts the helper T cells to scan them, right? Then these sort of T cells, transfers into or replicate into two types of cells. One is plasma cell, another is memory cells. Okay, this is the third phase. So let's review what happens in the first phase. In the first phase, the B cell finds an antigen which matches its receptor. At the second phase, you see it waits until it is activated by a helper T cell. Then the B cell divides to produce plasma and memory cells, right? in the fourth stage, what happens, you know? The antibodies are deactivating the bacteria. These are the antibody bonds, and from plasma cells, of course, they are killing the germs, the bacteria. And here, that one looks delicious. That means these eater cells prefer intruders marked with antibodies. That means these cells are very attracted with the bacteria which are attached with the antibodies, right? So this is the best thing they can scan for. Okay, and the last phase, you see, if the same inducer invades again, that means if the same germ enters our body again, the memory cells help the immune system to activate much faster. Yes, here. The response is a kind of secondary immune responses which is quicker and stronger than the primary response. Let's discuss something, related to this topic. It takes a smaller, stimulus to trigger a secondary response, and what is more, it occurs even after many years since the first exposure. Isn't it so amazing? Right. It takes a smaller stimulus to trigger a secondary response, and what is more, it occurs even after many years since the first exposure, as I told. Immunological memory is an important mechanism that protects the organism from bacteria, viruses, fungi, and parasites. It plays a significant role in understanding autoimmune diseases. It also is crucial in medicine therapy. The secondary immune response is dependent on many subpopulations of memory cells within T and B lymphocytes and natural killer cells. Here, I'd like to mention one more thing, that lymphocytes produce due to the activation of B lymphocytes and produces new B lymphocytes of similar category instead of producing the plasma cells. You must note it. Should I repeat this? Well, the lymphocytes produced due to the vaccination of B lymphocytes and produces new B lymphocytes of similar category instead of producing the same plasma cells. Clear? Well, these are so nice cells, examples of the memory cells: helper T cells, cytotoxic T cells, and T regulatory cells. Right, there are some notes related to memory T cells. It's written here. You may pause the video to look at this text very carefully. Okay, so memory cells live for many years or have the capacity to reproduce them. Second, the large number of memory cells are produced, and so secondary response is enhanced and is greater than the primary response, that is, memory against past infection. Number 3, they are activated by small quantities of antigens and require less stimulation than do the naive and unactivated T cells. Number 4, activated memory cells produce greater amounts of interleukins than do naive T cells when they are first activated. Okay. What about the fifth point? Comprise two subtypes. One is central memory T cells or TCM cells, and effector memory T cells, that TEM cells. Memory cells may be either CD4 plus or CD8 plus. These are important for MCQs though. Importance of memory cells in immunity. As you have already got to know that memory cells can reside in your body for lifetime even, and they are protecting silently without letting you know. So the principal functions of B cells is to make antibodies against the antigens perform the role of antigen presenting cells or APCs, and eventually develop into memory cells after activation of antigen interaction. On the other hand, T cells constitute 60 to 75% of blood lymphocytes. They can be distinguished from other lymphocytes by the presence of a T cell receptor on the cell surface. Another key surface of B cells and T cells includes the receptors it has in its surface. T cells recognize a linear sequence of amino acids, whereas B cells the spatial arrangement of proteins, nucleic acids, polysaccharides, or lipids. Okay, there is some important notes for helper T cells, cytotoxic T cells, regulatory T cells, and memory T cells. Just have a look, and I have already mentioned the importance in the previous lecture. Okay, so there are some more notes about the plasma T cells and memory B cells. Here is the diagram of the overall memory cell precursor, or this is showing the activation zone. Here, let's start the time over there. This is the activator or effector. When any germ is killed here, or they die here, okay, after the death they can produce the memory, and this memory cells and the previous some plasma cells, okay, what they tolerance they ask, or actually the tolerance level depends on their activation, and it takes a bit of time, okay. And this green color showing the adaptive or adaptotic cell, and this multicolored showing the memory precursor cells, and the red and green both are actually adaptotic or adaptotic cells, which are actually the combination of different sort of natural killer cells and plasma cells and sort of dead cells, okay. And here is the end of the lecture. From this lecture we got to know many things about the memory cells, the vaccination types of vaccines, and so on. Let's have some work at home, okay. You need to explain the roles of memory cells. Here you must mention what is memory cells at first, and you need to explain different types of memory cells and make some important points on the importance of memory cells. Thank you very much. Stay fine.